So now it's time to talk about the cell cycle. This is one of the last topics in the chapter, and the cell cycle is basically the sequence of events that happen in the life of a cell from when it's born to when it finally decides to divide and reproduce. There's four basic phases, and usually circular diagrams are used to describe them. G1, G1 is basically the first stage of the cell cycle after a cell has been produced. During that phase, generally speaking, G1 stands for first gap, but it is a growth period, and cells are manufacturing organelles, making lots of proteins and whatnot. Maybe arguably the most important part of a cell's life is right here. This is the checkpoint. That's the G1S checkpoint. At that checkpoint, the cell has to decide, do I want to reproduce again, or do I want to just exist? If it decides to just exist, it'll drop out of G1 and go to G0, and that's what the author's trying to show here. So at the checkpoint, it'll actually drop out and go to G0 if it decides not to continue on the cell cycle. If it decides to continue on the cell cycle, it will then proceed to the S phase, where it replicates its DNA. S stands for synthesis. And then it will go, without stopping, it will move into G2, which is the second gap phase. That's another growth period, but also major preparation for cell division. And then it enters the M phase. M stands for mitosis, where the cell actually goes about cell division. The periods of G1, S, and G2 are collectively known as interphase. So basically, you can also call the cell cycle as a combination of interphase and the M phase. The biggest checkpoint in the cell cycle where the cell can decide to either divide or not to divide is the checkpoint between G1 and S. There is a minor checkpoint between G2 and M, but cells cannot stop there indefinitely. They have replicated DNA, and they basically are destined to divide it. The reason this is called interphase is because it was considered like the period when cells weren't visibly doing anything. They're doing all kinds of biochemical stuff like cellular respiration, glycolysis, Krebs cycle, all that good stuff. But they're not doing anything you can see with a light microscope. Whereas the M phase, the cell actually divides in half, and you can actually watch that with a light microscope. As it turns out, most cells in a multicellular organism like us are not doing G1S, G2M. They're actually in G0. It turns out that G0 is maintenance mode. G0 cells actually perform organ functions. A kidney cell in G0 does kidney things, a liver cell in G0 does liver things, and muscle cells in G0 do muscle things. Cells that are doing G1S, G2, and M, they won't actually perform any organ functions. They just divide. Ultimately, they're doing the G1S, G2, and M to do cell division. So most cells in a multicellular organism basically are in G0 doing their thing. Cells that actually lose control of the cell cycle because of a damaged checkpoint become what we call cancer cells. Those cells are actually going around the cell cycle. They're not in G0. They don't do organ functions, and that's why cancer ends up being fatal. Because the cells don't do organ functions, they won't help a multicellular organism stay alive. So in that way, cancer is fatal. White blood cells generally are responsible for moving cells from G1 to the S phase. So this next part talks about cyclins. Cyclins are groups of proteins produced by genes that determine whether cells will proceed past the checkpoints, especially the major checkpoint between G1 and S. Basically, cyclins are a part of what is called the second messenger GPCR, G protein cell receptor communication systems. We'll talk more about those in the communication section of the third quarter. We're not going to be spending a lot of time on that right now. But basically what happens is white blood cells will move into an area that's been damaged. They will release chemicals that will cause the production of cyclins inside of cells that are in areas that have been damaged. The cells with the cyclins inside being produced then are stimulated to come out of G0 and go into the S phase to replicate the DNA and then to prepare to divide in G2 and then go ahead and divide in M. And that'll happen as long as the white blood cells are producing chemicals to stimulate intracellular cyclin production inside of the tissue cells that are dividing. Ultimately, that's how cancer cells get tissues to repair themselves. Activated cyclins bind to cyclin-dependent kinases in the cytoplasm, and that's how, and ultimately, the cyclins cause the cell to go through the cell cycle because of protein phosphorylation. It's very common. That's a common way of making proteins active or switching them on, is to stick a phosphate group on them. They call that phosphorylation. And G1 and S cyclins promote cells to go past the checkpoint and initiate DNA replication. S cyclins directly induce DNA replication. M cyclins help cells get through cell division. So ultimately, cyclins are really important to cycling cells, but not to keep cells in G0. And that's generally stimulated by immune cells. Now, mitosis is the process where cells actually go through the M phase. And mitosis is typically called normal cell division. Mitosis is division of one nucleus into two nuclei, followed by the separation of the cytoplasm to separate the nuclei into separate cells. The way it works is, and I guess we'll look at this animation here. We have a non-dividing cell here, and then what happens is, thankfully, the AP test no longer stresses the terms prophase, metaphase, anaphase, telophase, cytokinesis, but you're expected to know what's happening. And in prophase, cyclins are stimulating, M cyclins are stimulating the cell to actually supercoil the DNA into distinct chromosomes that each consist of two chromatids tied together by a centromere. So each X is actually two pieces of DNA tied together by a knot. The knot is called the centromere. Protein microtubules form. Those are formed from what are called tubulin proteins. They're actual tubes. They're not ropes. They're not solid. They're actually built like hollow tubes. And they radiate from the edges of the cell to the middle. The centromeres attach to the spindle fibers, and the spindle fibers are responsible for moving the chromosomes to the middle of the cell. At this point, the chromosomes are poised to pop apart. And I want to emphasize that DNA replication has made these two what are called sister chromatids in the chromosome identical. So this piece of DNA is identical to this piece of DNA. And this piece of DNA is identical to this piece of DNA, and so on. So when the centromeres break because of the release of calcium in the middle of the M phase, the sister chromatids are poised to be pulled to opposite ends of the cell. So this chromatid's identical to this one because of DNA replication. This one is identical to this one. This one is identical to that one. This one is identical to this one, and so on. So what we have here is two identical sets of DNA. This set of DNA is identical to this set of DNA, and these two cells then are clones. In the end, what happens here is that the cells actually continue to pull the sister chromatids apart, and then generally speaking, the sister chromatids unravel on opposite sides and two nuclei form inside of one cell. Each nucleus is said to be diploid because it still has, and you'll see this in the meiosis animation, it still has ultimately chromosomes in pairs. Then cytokinesis occurs and the membrane pinches in half, and you end up with two cells formed from the original one cell. This is a cloning process, and that's how the body replaces missing cells. Meanwhile, this is, that was animal cell mitosis. Plant cell mitosis is very similar. In this case, we have a plant cell, which tends to be usually a rectangular or a hexagonal shape. and the cell actually does the same basic thing, forms a spindle, it gets rid of the nuclear membrane, it attaches the centromeres of the chromosomes to the spindle, the chromosomes line up, the centromeres break due to a calcium release in the middle of the M phase, the sister chromatids are pulled apart, two new nuclei form, but then instead of pinching in the cell, because the plant cell can't with this thick cellulose membrane on the outside, they build a plate on the inside out of the spindle. And then in the inside of the plate, they build new cell walls and membranes, and then you end up with two separate cells that are completely distinct from one another. And they're also the same as before. They're what we call, they're diploid cells. They're clones of one another. Now, meiosis is a divisional process that's different. Now, meiosis only produces sperm and eggs. The sperm are actually made slightly differently from the eggs, but in the end, the two processes end up similarly mixing up DNA instructions so that the offspring cells will be different from one another. And in this case, the way this works is the meiotic cells actually go through two separate cell divisions in a row. In the first cell division, they divide what are called the homologs. And in the second cell division, they divide what are called sister chromatids. Now, I'll go through the text here, and then I want to show you the animations that kind of explain everything. Basically, meiosis is two consecutive meiotic divisions in a row. The first division is by far the most important. It allows the mom-dad pairs of chromosomes to be separated. That is important because if that can't happen, diploid organisms cannot have successfully, at least they can't successfully make offspring. So this says the exchange of information between mother and father DNA in the first division is called crossing over. Meiosis guarantees that no two children born from separate sperm and egg will ever be clones. Meiosis makes sure that all eggs manufactured by a typical female are all different. No matter if it's 20,000 or 100 or 50, all of them will be different because of the way the DNA is swapped between homologous chromosomes. So crossing over tends to be rather random in DNA swapping, and then depending on how the mom-dad chromosomes are separated, that'll determine the distribution of DNA into the sperms and the eggs, and that'll determine that they not be clones of one another. Basically, the second meiotic division basically just separates the centromeres of the chromosomes so that individual chromatids go into the eggs and the sperm. So at the start of meiosis, cells are diploid, but at the end of meiosis, they're haploid. And haploid means there's only one set of instructions in each one. The genetic variety of meiosis makes sure that offspring have the best chance of surviving on a planet where conditions are always changing. And now we need to go over sperm and egg meiosis. So, let's go back here. And I'm guessing I got to get to sperm production here. This is here's the sperm. Okay, so let's look at it here. Here's looks like an egg in a frying pan. It looks just like a mitotic cell, but something strange happens at the start here. When the chromosomes form up and the spindle forms up, the nuclear membrane disappears, something strange happens, and the chromosome pairs find each other. This is a diploid cell because there's two blues, two yellows, two orange, and two polka dot, black and orange. The chromosomes are in pairs because these are mom-dad sets. If this is a paternal chromosome, that's a maternal. If this one's maternal, this one's paternal. The DNA came from parents. And right now, in the meiosis of this organism, the DNA is being sorted. This sorting process is critical because not only is it essential for the chromosomes to be distributed to the eggs or the sperm, in this case, properly, but this allows crossing over so that the chromosomes will trade pieces and guarantee that they will not be making identical sperm in the end. Then the homologs, these are called homologous pairs. homologous pair, homologous pair, homologous pair, homologous pair. They line up and they separate. No centromeres break. And the homologs are pulled apart, and then the cells literally split in half. And then without even unraveling any DNA, the chromosomes line up again. And these sister chromatids in here are no longer identical because of crossing over. The centromeres pop due to calcium release, and then the chromosomes are pulled apart, and then the cells split in half. You end up with 4 sperm from the original cell that started meiosis. And now we need to look at egg meiosis a little different. So let's click on this, and this will come here. Egg meiosis is different in that the egg has to be really big and not tiny like sperm, so that the egg has the nutrients needed for fertilized or for an embryo to survive. So in this case, it looks the same at the start. The nuclear membrane disappears, the chromosomes supercoil, the spindle forms, and the synapsis occurs. DNA is crossed over between the homologous chromosomes from mom and dad, and then the chromosomes line up, and then no centromeres break, the homologs separate, but then a polar body forms. So this polar body over here is completely gypped it's shorted of cytoplasm. It will die. This is called a secondary oocyte here. Now, this is the cell that human females have in their ovaries. Women actually don't have eggs in their ovaries. They have secondary oocytes. The sister chromatids have not been separated inside of this nucleus. If a sperm was to reach a secondary oocyte at ovulation, the nucleus will complete the second meiotic division, and that will allow then the sperm nucleus to join the egg nucleus and form the fertilized zygote. So if a sperm is actually against the egg membrane, that will trigger the second meiotic division. The second polar body will form. This polar body will die, and then a little sperm nucleus then will be pulled into the egg nucleus, and then the first thing the embryo does is it replicates its DNA and prepares for the first cell division. It goes through the cell cycling, except it skips G1 and G2. The egg, that's one of the beauties of the egg. The egg is big, it's full of nutrients, and it doesn't need to do G1 and G2, and this saves the embryo time in early development, and that's pretty important. So let's go ahead and close that. Let's go ahead and close this, and let's go back to the original notes. So the crossing over process, this illustration kind of helps to show this is a cell at the start of meiosis. It only has 2 chromosomes. And then here it is at the end of the first meiotic division. This cell has separated the 2 chromosomes. But you'll notice the red chromosome has a blue piece on it, and the blue chromosome has a red piece. That's because of crossing over. And then in the second meiotic division, you end up with 1 chromatid in each cell. And all 4 of them are different because of the crossing over that happened here at the beginning. So even though the red chromosome may be maternal and the blue chromosome may be paternal, the 4 chromosomes in the 4 cells are all different because of the way they exchange DNA and separated. That allows meiosis to make sure that there will never be cloned babies born from separate sperms and eggs.